conduction velocity because of normal coupling across the transmural direction, then you almost don't see gradients. It takes uncoupling of whatever fibers that are represented across the myocardial wall to start seeing, and then it surfaces, and of course, when you have a very high degree of uncoupling, it approaches the single cell differences. And the M cells that we put in the middle there were based on low expression level of 5Ks, which is known to be the case for Charlie's study. But for me, I think we all have to be careful with what the models can do and predict and their limitations. If you take Charlie's wedge, that model is chemically generated low QT syndrome or Brugada, right? It's pharmacologically. It doesn't have structural abnormalities. It takes a normal dual heart, no fibrosis, no structural heart disease, and then makes it to be a Brugada or a low QT. So of course you cannot find production abnormalities because of structural abnormalities, because they are not there.
So if we agree that there probably isn't transmural gradients in the long QT, because they're structurally normal, what explains the gradients that you see? That's what I'm trying to struggle
We
with.
see the gradients we see on the epicardials are much greater than the gradients that you see on the corpuscle.
What is your concept? Why do they have
Why do we have those gradients?
Yes, in the long QT.
Okay, so that's what I was trying to explain to, or trying to say to Colin. So, long QT syndrome, let's take any one of them. It has a mutation in an ion channel that causes prolongation of the action potential. Now, let's assume, and this is not a good assumption, but let's assume that this mutated channel is now expressed in the heart uniformly everywhere.
Okay.
But the substrate to begin with is very non-uniform. There are many differences in, for example, expression levels of IKR and other channels. Now, it takes very small change to change the action potential by a lot. So, if you have a homogeneous expression of the mutated channel, even when it's not a good assumption, but let's assume it's uniform. On the background of non-uniform expression level of other repolarizing channels, you affect the action potential very differently in different places.
Okay.
Now, a better assumption is that even your expression level of the mutated channel is not uniform. And then in many of these hearts, you probably have some structural changes and uncoupling and situations like that. So why expect it to be homogeneous?
Well, I guess I was trying to argue with that, saying that with the Buehler-Reuter model that we use, that you could get cells very close to each other with very different action potentials, just dynamically you could get that.
Minute to minute, you mean, beat to beat.
Right.
Yeah, I mean,
Right, because it's stochastic, right?
yes. Could happen, and then it gets to some steady state.
The interventricular septum wedge preparation is a cut surface, and the cut surface is biophysically a reflection barrier. So that affects the electrotonic interaction. It does
We
not
see a lot-
replicate what you see in a whole heart. It is better than nothing, but it has its limitations. So also there, one has to be very careful with the interpretation of
But
the
in many situations, we do see, interestingly, like in non-ischemic cardiomyopathy, we see slowing of conduction across the septum, or we see the gradient of repolarizations across the septum. And this is because, yes, locally, as you cross the septum, you have large changes in load. And the wavefront, if it's a planar wavefront, goes across the septum. Now it has to divide into going into the septum and from the septum. So you have, it's basically an expansion.
Exactly.
I mean, you did experiments on bifurcation and expansion. That's the same phenomenon. Any other comments? Well, if not, I think it's been a long day of science. That means thank you for being here. This is really the first CBAC seminar of the fall semester. And it really follows what I thought was a fantastic symposium celebrating our 10 years of existence at the end of August. So really one good thing leads to another. And I really couldn't think of a better opener of our fall series than Professor Bertil Hille, who's here. He was kind enough to come and share his time with us all the way from the Northwest, that is Seattle, University of Washington. Sounds similar to Washington University, but So Professor Hille really doesn't need an introduction. He literally wrote the book on ion channels and really developed in a pioneering effort many of the concepts that we know today and we take for granted as the structure of ion channels, such as the pore and how the pore is open through voltage-gated movement of a voltage sensor. And these are concepts that were developed by Professor Hille. So without further ado, and I can't really count and list all of the awards that you received, but I should mention the Lasker Award among many others. And without further ado, these are your captive audience.
Okay, well, thank you very much. I've had a wonderful day. I'm going to stand here so that you won't be able to see. Oh, you have some work with you.
I'll tell her.
Okay, so today I'd like to tell you about the contents of this list, actually. So, since I'm assuming that there's a varied audience and some junior people there, I'll try to be didactic and simple and understandable throughout. So I'll tell you or remind you about GPCRs, the G-protein-coupled receptors. I will remind you about inositol phospholipids. And I'll remind you that phosphoinositide bisphosphate has interactions with ion channels. So those are all reminders because there are world's experts in those areas right here, and you've probably heard a fair number of seminars already about them. And then I will go on to the biophysics and the cell biology of lipid signaling. So let's, without any further activity, this is, by the way, P2, but we'll come back to it in a moment. Let's go on to the G-protein-coupled receptors. Again, in a didactic manner. G-protein-coupled receptors are orange, 7 transmembrane proteins that are exposed to the outside, a region which is a receptor, and the receptor is the recipient of a whole variety of neurotransmitters, peptide hormones, and sensory inputs, different receptors for each one. And when on the inside, the G-protein-coupled receptor is coupled to a heterotrimeric G protein, which has, as you can see, alpha, beta, and gamma subunits, and the alpha subunit has GDP in the resting state. But when it interacts with its activating receptors, which bind either in little pocket down in this region inside the membrane, or if it's a big peptide right outside, then something happens. The receptor changes conformation. It interacts with the G protein in a new way. The alpha subunit actually sticks right into a gap in it and catalyzes a change in the alpha subunit, so the GDP departs and GTP comes in. And then in the classical textbook description, there is a separation of alpha GTP from beta gamma from receptor. So you have now 3 intracellular messages that are bound to the membrane by lipidation and by transmembrane segments. So I said this is the classical textbook view. Now, to just touch on a few of the things that are happening in the field that are still not totally resolved. Many of these receptors act as dimers, so two of the receptors together. They can be heterodimers, so you can have two different agonists. And when they are heterodimers, they may have a new pharmacology and a new signaling possibility, and even coupled to different G proteins. Some people would describe the receptors as sitting with a G protein on them, all ready to go, and others would think of the G protein as mostly coming afterwards. But in any case, the G protein, once you have the active form of the receptor, multiple G proteins come one after the other and are activated sequentially, making amplification. Gautam in this university has shown that beta-gamma subunits appear not only in the plasma membrane, but in all kinds of intracellular spaces and membranes. Some people feel that in some cases the receptor does not dissociate from the alpha and beta-gamma, that they still stay together, but they just change their conformation, and in fact even the effector is there with them, and so you have a really fast, short pathway for a signal. And I think probably all of those things are correct for some cases. And just to say that the textbook description is kind of the first descriptions which came from rhodopsin and from beta-adrenergic receptor, and now things seem to be, there are some variations on this theme, which is always makes it nice in music, they have variations. So let's go first, since we are in a cardiac environment here, we'll mention a cardiac example. So here, everybody knows that when you exercise, you stimulate sympathetic nerves, noradrenaline comes out, beta-adrenergic receptors, and this couples to the G protein GS, from which the alpha S subunit then activates adenylyl cyclase, cyclic AMP dependent protein kinase starts to phosphorylate. And in the case of the heart, we know of at least 20 different proteins that are phosphorylated, so a large number of different targets, many different things. They all accomplish the same thing. They're all going towards a greater cardiac output. So they all are the things that you would expect to happen when you want to have that as your final output. This recording is from the pacemaker, and it shows the pacemaker speeding up. By contrast, if you stimulate the vagus nerve, acetylcholine comes out, the M2 muscarinic receptors are stimulated, GI is the G protein, and it's the beta-gamma subunit coming from GI that acts on an ion channel, a potassium channel. See, they are lots of different effects, but I'm just focusing on a potassium channel is activated, and potassium channels bring cells down to a more negative potential and hold them at rest, so the heart slows down. So there's an example of alpha and beta-gamma. We have lots of examples of beta-gamma and alpha, but there are also, I didn't mention it, the third signaling component, which is the receptor. The receptor, when bound to the ligand, signals not only to G proteins, but it is a scaffold for other signaling proteins. So there are three arms of signaling. Today I will focus only on the G alpha arm. Okay, so let's, continuing in this vein, how many of all these things are there in the genome, in the body? And so I made a kind of a numerology. How many agonists, how many receptors, and so forth. And I just made it up. I pulled it out of the hat. And there are thousands of agonists, especially if you think of flowers and perfumes and cheese and wine, all these odorants that make all these interesting odors, which are G protein coupled. But also there are hundreds of physiological agonists. Then there are in the genome 500 to 1,000 functional receptors. There are also some pseudogenes that interact with these agonists, and then they couple to only a small number of G proteins. So I should have said, I meant to say here, G protein families. Okay, here it says families. 4 families I will identify. There's a 5th family which happens in the outer segment of the eye, transducin, rhodopsin transducin interaction, but I'll discard that as all the rest of the body doesn't care about transducin. So we have only 4 families similar behaving G proteins. And so you might say, that's kind of weird, how do you have 1,000 signals, very interesting, rich environment, and you reduce it to 5? Aren't you throwing away all of this interesting stuff? And the answer, I would say, is that no cell has a great variety of things it can do. If you have a heart, it can speed up and slow down, kind of thing. The more cardiac output, less cardiac output. You don't have a variety. It's not like all the 64 million colors of pixels on those screens. The cell doesn't have that. So you only need 4 signals and the combinations of them to do everything the cell ever wanted to do. That allows the convergence, as it were. Then they make tens of messengers, 10, 20, 30 messengers would be known in the cell, diffusing in the cytoplasm and other places, and there are thousands of effects. Well, if one family can give rise to, let's say, 1/5 of 1,000, which is whatever it is, 200 effects, how do you, if I had adrenaline, will it make my heart secrete and produce endocrine stimuli and contract and do all the other things that GS can do? No. Each differentiated cell produces a smaller repertoire of the effectors. And so by producing the appropriate set of effectors, each cell can design, as it were, what is it that cyclic AMP-dependent protein kinase is going to be able to do. It's going to be able to phosphorylate some serine and threonine residues, but you restrict the possible outputs. So this is sort of my rationale for this enormous number of outputs, enormous number of inputs, all going through a very small number of vectors. Okay, so then let's just continue one last little bit about the vascular system. In the vascular smooth muscle, for example, I picked a couple of agonists, a couple of receptors that act on GS. They make cyclic AMP go up, and then you have relaxation and vasodilation and better perfusion of the vascular bed. On the other hand, you have another set of receptors and agonists, GI, less cyclic AMP, and they make contraction and vasoconstriction and less perfusion of the vascular bed. And here we have only 2 modalities. We had with 2 G proteins we've gone, so what is the range the smooth muscle in the vascular system is going to do? And this almost covers it. But there actually is one more set of agonists and receptors, and they go to GQ. They make IP3, calcium, diacylglycerol, protein kinase C. But the end result, again, is more contraction, which sort of complements this system here. This is an interesting example, though, because all 4 of these receptors also couple to G12 and G13, another family of G proteins. And those activate Rho and Rho A, Rho kinase, ROC, and they contribute to the contractile promotion of these other. So here we have a case where one Gproteincoupled receptor is coupling to several families of G proteins. In this case, they use different effectors, but they give the same end result. And maybe other cases would be even more interesting or more complicated. Okay, so I've now finished my compliments to the cardiovascular system. So I will focus on GQ, activated by muscarinic receptors in my case, where IP3 and calcium and diacylglycerol and protein kinase C are manufactured. And phospholipase C has as a substrate a lipid, the lipid that we are most interested in. So let's go to the neuron example that brought us to this field. Namely, here is a recording of time and voltage from a sympathetic ganglion cell, neurocerebral ganglion in the neck. And it is being depolarized by a current pulse, and when you do that, it fires. It doesn't fire too well. It just fires a few times. And the reason it fires only a few times is there is a potassium channel which is open at rest, which keeps the cell from depolarizing to the threshold, which is this green line. And if you close the potassium channel, you could do better. So if you add a drug that closes the potassium channel, then now the cell depolarizes a little bit, it reaches the firing threshold many times, and it goes. So it's actually a very good follower. It's awake, it's paying attention. But the interesting thing that brought us to the phenomenon I'm going to talk about is that you can do this with a Gproteincoupled receptor. And that is system. Okay, good. So here, instead of using a drug to block the channel, we're using an acetylcholine mimic that acts on muscarinic receptors. And the muscarinic receptors have turned off the potassium channel. This is a receptor that goes through GQ. Okay, so I should mention that KCNQ is also quite closely related to KCNQ1, which is the repolarizing delayed rectifier of the heart. It's in the KV7 family. In the case of the neuronal one, if you have genetic defects, down loss of function, you may have epilepsy, which could be expected when it's in the central nervous system, as in it is in the hippocampus, for example. Then you'll have excess firing compared to otherwise, because the potassium channel is not restraining the cell from firing. And in the case of the heart, if you have KCNQ1 downregulation, then you have a long-action potential, which gives rise to long QT and possibly sudden death. All right. How does it work? What is the receptor doing? That was the problem that led us to what I'm going to tell you about. And the answer is, namely, that the ion channel, KCNQ, all the KCNQ family, likes and insists on having PIP2, a lipid, as a cofactor. And without the PIP2, the channel is asleep. And when you activate receptors coupled to GQ, then phospholipase C acts on PIP2 to make these interesting messengers. Yes, indeed. But the interesting thing in this case turns out to be the loss of PIP2. So not only do you make things, but you lose some. And the loss causes the channel to go to sleep because there isn't a PIP2 that it needs to stay awake. All right, and so that makes the channel wake up, and the channel go to sleep, and the cells wake up. Okay. So let's now start investigating the system. We consider first the substrate of phospholipase C, which is a phosphoinositide. Phosphoinositides are conventional phospholipids, 2 fatty acids, glycerol, phosphate, a head group. But in the case of phosphoinositides, the head group's name is inositol, myoinositol, a sugar-like molecule, lots of hydroxyl groups, which presents to the cytoplasm its 3 and 4 and 5 hydroxyls to be phosphorylated by lipid kinases and lipid phosphatases. And when that happens, you get all the combinations you can expect. You start with the unphosphorylated PI. You make 3 different ones that are monophosphorylated, 3 that are doubly phosphorylated, 1 that's triply phosphorylated. And the one we commonly call PIP2 is PI45P2, even though these other ones also have 2. But so I'll try to say 4 or 5 sometimes, but most of the time I'll say PIP2. So let's consider again the activating GQ and phospholipase C. Phospholipase C will cleave this molecule at this point, making IP3, diacylglycerol. What are the kinetics? I'm a biophysicist, so I want to know how fast and what the reasons are and all that kind of thing. Oh yes, let's first show there was doubt in the field that the agonist could actually deplete the lipid. Maybe a little bit, but a lot, that was not commonly considered. So we had to test that. And here I'm showing you now unpublished work in sympathetic ganglion before we have done it in cell lines. So we have the 3 lipids that I mentioned. We have 3 lipids. PI, which makes PI4P, which makes PI45P2. And if we activate a receptor, it should act on PI45P2. Down below, we are measuring with mass spectrometry the amount, the mass of the lipids in the whole ganglion. So we can't do it in single cell, so we have it in the whole ganglion. So it says that the PI is about 96% of all the phosphoinositide. And the PIP and the PIP2 are each about 2% of total phosphoinositide. And now the PI itself is a relatively rare lipid compared to the other PCs and PS and all those other ones. So this whole group is a low abundance group, very low. Their roles are not really to make the structure of a membrane. Instead, they are instructional. They are information that informs proteins in the membrane and in the cytoplasm. And we'll see about that. Okay, so now you activate the receptor, and let's look at this bar here. This bar is supposed to be 2% of PIP2. So I will activate the receptor and for 60 seconds, and clearly the PIP2 went down in the whole ganglion, including every kind of cell that's in the ganglion. And the PIP went down too. So this was good for the hypothesis, and this was something that was thrown in addition, and so then you have to realize that actually the PI4P pool is, sorry, did I go too fast there? The PI4P pool is in rapid exchange with the PI45P2. And so if this PI45 is being depleted, then the PI4P is supplying it, and it is depleted. Also, Philip Majerus in this university showed maybe 30 years ago that phospholipase C could actually work a little bit on the PI4P, so that's another reason that PI4P goes down. Okay, so let's consider how fast is this? How long does this last? And the answer can be seen in this kind of experiment, first of all. So here we're measuring the potassium channel, KCMQ. Measure, it's all controlled and stable. We add the muscarinic agonist for 20 seconds, 4, 8, 12, 16 seconds it takes for the current to go off, and that's how long it takes for the lipid to go down too. And then you take off the agonist, and now it takes 100 seconds for the lipid to recover. So there's a period of 100 seconds during which this cell will be hyper-excitable. You just have now turned off some of your KCMQ channels, and you're now listening a little bit better. But in 100 seconds, don't feel relaxed. So it's interesting to us. Why does it take 100 seconds? How does it take 100 seconds? And what sets the timing? Is it something you can vary? And so those are the kind of mechanistic questions that I will now consider addressing. Okay, so during this period, okay, the lipid is being depleted, and during the recovery period, this synthesis from PI, because this pool is more or less gone, this is gone, this whole synthesis has to take place. So we measure the rate constants for everything on the graph, including the diacylglycerol and the downstream products. We've made a model of all of it, which I will not tell you, but how do you measure? So I would like to tell you about how we do measure some of these things. So I give you the example from the thesis of Jill Jensen, who wanted to measure in her thesis the rates of all of these steps: receptor, G protein, phospholipase C, PIP2, and channel. How did she do it? She used fluorescence resonance energy transfer, which is called FRET. And in each of these places where there's a colored circle, she put either a CFP or blue fluorescent protein or a YFP or yellow fluorescent protein. And when CFP and YFP are near each other, there's a transfer of energy from the blue to the yellow called FRET. So you can measure when molecules are near each other. So this is done pairwise. So let's consider receptor and G beta, for example. That's one example of one of her sets of experiments. How long does it take, you add agonists, how long does it take for the beta subunit to know that the agonist is there? So her experiment was done as follows. CFP in the C terminus of the receptor, YFP in the beta subunit. If you shine blue light in, out comes blue light, unless YFP is nearby, in which case blue light will transfer energy to the YFP and out will come yellow light. So then you measure as a simple measure, YFP divided by CFP, which is a fret-like measurement. Here's 1 second in time, divide to 6, and it shows that in 200 milliseconds, the beta subunit learned that agonist had been added to the back. Of course, you have to have fast flow and quick applications. And it takes about 4 seconds, notice the time scale is different here, for this to be forgotten. And in this way, she marched through the different components, and I'll show you the summary of all that, which is here. I notice that the time scale is logarithmic, so in order to represent quite a number of different times. The one that we just looked at is 200 milliseconds it takes for receptor to talk to G beta. About a second it takes for the phospholipase C to learn from G alpha Q. Those are the fret pair. And even though the phospholipase C then would be active at this point, it takes another 10 seconds for the PIP2 to be destroyed and the channel follows right away. Why does it take 10 seconds? Well, there is endogenously in the cell, there's a certain amount of phospholipase C, and there's a lot of PIP2 substrate, and it just takes that long for the system to grind through the system, through the PIP2 that's there. If you overexpress phospholipase C, which we could do, we could change this from 10 seconds down to 2 seconds, so you could show that that was the rate limiting step in that case. And that we did that for many different steps to show that they are rate limiting. Now at this point, I have a choice of shortening my talk or making it as much as I said I would. And Dr. Rudy is saying, last night he said, oh, you can go for an hour and a quarter if you want to. So that encouraged me that I might actually speak for a whole 60 minutes, if that's all right with you. So then I will include the slides that otherwise I would have skipped. So we made the model, as I said. So we now have a mathematical representation of everything, which describes quite well the observations that we have. And so I want to now think about what pharmacologists call the concept of spare receptors, what we might call as dose response curves, and they might more accurately call concentration response curves. What sets the rules for that? So the binding of agonist, oxytryptamine, takes 2 micromolar oxytryptamine to get 50% occupancy of the receptor, the M1 receptor. Then that's the first reaction here. But then you have phospholipase C being activated. How much agonist do you have to add to activate phospholipase C? I'll leave that as a slight thought question for a second while I take a drink of water. Okay, and the answer is much less. So here's the activation of phospholipase C. So that tells us right away that with receptor systems like this, which have a cascade, the downstream events don't follow, don't indicate to you the binding percentage. They are normally, need less than the half binding. And I'll show you that more in space. So what that means also, as pharmacologists would say, there are spare receptors. You only need to activate this many receptors to get half the phospholipase C. Now the phospholipase C is endogenous, but the receptors are overexpressed. This is now, I'm not talking about a neuron here, it says TSA cells. When you see the word TSA cell, as you did actually in the Fred experiments, I forgot to say that, we are expressing the components in a cell rather than using only endogenous components. So, we naturally would say, okay, you put in too many receptors so you don't need them all. Way to go. Okay, so let's go down. Diacylglycerol IP3. Wow. You need less and less and less. So to make diacylglycerol and IP3 from PIP2, this is the curve. You're down, what is it, a factor of 100 almost. It just means that you really don't need anywhere near the endogenous amount of phospholipase C to generate all the PIP2 and all the IP3 and all the diacylglycerol that you can generate. Maybe they come out a little slower, but they all come out. And then calcium, wow. That means that you only need maybe 1 or 2% of the total IP3 that you could make to make a calcium signal is significant. It means the IP3 receptor has a very high affinity for IP3. So this is remarkable. I think this is all these. These are all drawn from the model, but they all are what the data say. So that's just a comment on pharmacology. Now you say, okay, that was too many receptors. And this is the curve for too many receptors. We actually have 500 receptors in the model. The cell has more like 20 receptors, so it's factor 20 or lower. And let's go all the way down. Let's go 400-fold down. So let's take an example where you have 1.25 receptors. So now we're at the lower level than the neuron was for its endogenous muscarinic receptors. What happened? Well, all the curves slide towards the higher concentration. You need more concentration. But look here, what's this? IP3. Here's IP3. Compare that to IP3 over here. It doesn't get above a little bit. So now you would say, if you just had this experiment, you'd say that means that oxytremerine is just a partial agonist. It didn't make the full effect. Actually, what we needed was more receptors, then we would have had the full effect. So this is all to say that with different receptors and different receptor numbers, which we have in cells, you might see very different effects, even though they're using exactly the same downstream system, and they work exactly the same way as the model does, work exactly the same way. And you might say, oh yes, the other agonist, for example, in our case, ATP also activates GQ, and it does more or less what this picture. And you'd say, oh yes, ATP must use a different mechanism because it's so different from the other one. And the answer is no, it's just a different density of receptor. Okay, so that was one of the asides that I could have thrown away. Let's go on to another method for working with other channels and working with PIP2. And that is the so-called voltage-sensitive phosphatase. So Okamura found a molecule which had a voltage sensor, S1, 2, 3, 4, with lots of positive charge, exactly like voltage-gated sodium channel, potassium channels, and calcium channels. But then it had not a channel. It had an enzyme, very convenient enzyme for us. This is an enzyme that will convert PI452 to PI4P. And all we have to do, we are electrophysiologists, so we love this, is apply a voltage for 1 second, and then we have converted the PI452 to PI4P. So with this kind of tool, you could express any channel you like in a cell, and this voltage-sensitive phosphatase, and then you could ask, does it care about PIP2? So it's very handy that way. So we did that for 10 of the, or you tried 10 voltage-gated calcium channels. Here they are, all listed, their numbers. The 2 yellow ones are ones that are prominent in the heart, and 4 of them, 1, 2, 3, 4, are prominently affected by PIP2. So this is a way of screening, and it also tells us that there are many channels in the cell that care about PIP2. If you go on and ask how many, then you can get a list that has 80 ion channels, all the inner rectifiers, including the KACH, as it's called in the pacemaker region, the one that hyperpolarized with acetylcholine, all the KCNQ channels, all the TRIP channels, and members of all of these families, and also 6 transporters in the plasma membrane. These are all plasma membrane, 6 transporters care about PIP2. The pioneer in this area was Don Hilgeman, who's from Texas, and he showed that an inward rectifier called KATP channel is sensitive to PIP2 in the heart. That was the first time anybody said that kind of thing. And the sodium calcium exchanger in the heart was also. So we have to pay homage to Don for this actually fantastic insight, and I'm going to tell you another one of his insights. But now, we're going to use KCNQ from now on just as an indicator of PIP2. So we'll just say KCNQ is my indicator of PIP2. If you hadn't heard it before, you might say, well, why should all these different channels care about PIP2? What biological good would this do? And I'll now discuss that with respect to a Don Hilgeman hypothesis, namely that if this is a familiar diagram of a cell, endoplasmic reticulum, Golgi, vesicles, and going in again. And some of the membranes are colored different colors because some of them have different dominant phospholipids. So here, bright green is the PIP2 we were talking about before. But in each of these other membranes, there are different lipids, which are dominant phosphoinositide lipids that are kind of markers of those places. They're zip codes, as it were. Now, the first question you might ask is, okay, we know there's a lot of traffic here, so the lipids must traffic too. Why don't they all get mixed up? That's a very good question, and the answer is, for example, Pietro de Camilli showed clathrin-coated pits form as diagrammed here. The adapter and the clathrin like PIP2 and come down as they do here, and dynamin comes in liking PIP2 and it squeezes, and synaptogenin, a molecule, comes in and it's an enzyme that takes off the 5-phosphate. So all during that process of forming and pitching off a vesicle, you're ready to take off the 5-phosphate so that now this intracellular vesicle no longer is PIP2. And that happens, we would say, at every one of these junctions. So there are phosphatases and lipases that work, and I would say it all happens in 1 second. The enzymes are powerful enough so that in 1 second you change the color of the lipid, and so you don't have lipid mixing because you just paint over the old one and make it a new one. The striking change. All right, so now we're trying to get to Don's hypothesis. So, what happens to channels and transporters? Channels and transporters being made here. They traffic all this way. Don Hilgeman pointed out that they don't see PIP2. They only see PIP2 out here, so they would be off. And that would be a convenient thing for solving a physiological problem of why doesn't the sodium-calcium exchanger start working from the endoplasmic reticulum? It's all in these transport vesicles and in here and do all kinds of bad stuff to the physiology. And the answer, according to the hypothesis, which is still a hypothesis, is that the lipid is actually regulating whether those membrane proteins are able to do their job or not. So a test of that hypothesis is to, for example, look at intracellular vesicles and to patch clamp them and see what they do in terms of channel. The method for that was invented by Hausch and Shu in Michigan. Namely, and he was working in David Clapham's lab originally when this was discovered. Namely, he was able to fuse together what he called endolysosomes by adding a small molecule drug called vacuolin, and I'll show you pictures from the work of Eamon Dixon. Now, Eamon Dixon should be familiar to all of you because he's applied to at least 2 jobs at this university in the last few weeks, and he's an extraordinary scientist, so I call your attention to it. So here, Eamon Dixon has expressed KCNQ, YFP, and we've taken a picture confocal and turned it into a negative, so the dark is with a KCNQ. It's 24 hours after expression, before the KCNQ has finally trafficked to the cell surface. At 48 hours, it's all on surface, but at this time, it's intracellular, in something, in vesicles. And if you add vacuolin, you see these bubbles starting to form, and they get bigger and bigger. This is all the same cell. They just fuse a whole bunch of stuff. And then you can take the shell and you can shake it, and out come the vesicles. You put it in intracellular medium at that point. And now you have vesicles you can patch clamp. This is what Hausch and Shu originally developed. And Eamon was expressing now the KCNQ channel. And then he did patch clamp. So here is the voltage protocol going up in a ramp and down. And the ramp is going in time. So time is here. That's just going up the ramp. And you can see that there are currents, some channels present in this vesicle. If you add PIP2, then move up to the green line, you have more current. And if you add the blocker of KCNQ, you go back the same amount to this red line. And so this seems to show that there are KCNQ channels that are functionally capable that will turn on if you give it PIP2 in these intracellular compartments. So that's an example of trying to test the Hillebrand hypothesis. Okay, so let's move along to other ways of changing the phosphoinositides. So now we're becoming very phosphoinositide oriented and almost forgetting channels, except they are a good indicator. So Gupta Dai in my laboratory was thinking about how PI was phosphorylated once, phosphorylated twice to make PIP2, and that PI came from myoinositol, which is added to D8 diacylglycerol to make the PI. And I asked them to overexpress a transporter which brings myoinositol into the cell. So the sodium-coupled myoinositol transporter takes sodium in down its concentration gradient and brings myoinositol uphill. So this is a way to bring myoinositol into the cell. So what would happen? What would you expect would happen? Well, he thought, this should enrich this pool of intracellular myoinositides. So he measured them with the mass spec, and here in black is the PIP, and here in black is the PIP2, and sure enough, they got vastly bigger when you overexpressed the transporter, and also gave extra myoinositol, and this is for 24 hours ahead of time. If you only give myoinositol, in fact, you also pick up the amounts of PIP and PI45, PIP2, but the transporter actually is very helpful for giving more. This transporter is closely related to the glucose transporter. It's the next thing in the same family as the glucose transporter, and it is called also a osmolyte transporter. An osmolyte transporter brings in osmotically active particles to adjust osmotic pressure. And in fact, if you put the cell in hypertonic solution, there is a transcription factor called TONS or tonicity that turns up the expression of SMIT1, and you actually increase the amount of myoinositol inside the cell as a kind of a compensation for the extracellular tonicity. This would happen, let's say, in the kidney, where there are different osmotic pressures, and maybe also in the case of drought, where you want to have a drink. Who knows? But in any case, that also boosted the pools of PIP2 and PIP. So there are physiological ways that can change the pools. Now, what would you expect would happen when you increase these pools to the regulation of ion channels? So he asked that question, and so now we have bigger PIP2, bigger PI PIP2 pool, bigger PIP pool, and he found that there was slower suppression by oxytremerine, this receptor, of KCQ channels, TRP channels, which also depend on PIP2. So it was harder for this system to deplete the pool. It's a bigger pool. And after depleting it some, there was much faster recovery after oxytremerine or after the voltage-sensitive phosphatase of the KCQ channels, or ERK2 channels, which depend on PIP2, pH domains that depend on PIP2. They all were more quickly replenished, as if this pool is helping the whole phosphoinositide enlargement is replenished. And interestingly enough, when you have a larger pool here and you activate phospholipase C, you make more IP3 and you make more diacylglycerol and you make more calcium. And I'll show you that measurement. But the bottom line is that the cell's excitability has changed a lot. So it's harder for me to get the lipid down enough to turn those channels on. And then they pop right back again. So it's kind of like narcolepsy. I mean, you don't really pay attention. That's all right. Let's leave now. Okay, so let's see the effect on IP3 and calcium. Here's a measurement of IP3 and a measurement of calcium as a function of time after oxytremerine. So the black line shows that IP3 goes up and down with a FLEP probe, and calcium goes up and comes down with a Pura probe. And after SMIT1 and myoinositol, this is larger, probably it's saturated here, so it's probably larger and longer, and so is the calcium signal. So all kinds of signaling are different, just from hypertonicity or from overexpressing them. So that shows that there are physiological regulations of these systems, which we've studied for such a long time. We sort of thought, well, if it do is some kind of constant, there's a regulator that sets it. No, it actually is variable. Let's consider about another way that it might be variable or varied. I talked about this scheme up here. This is a simple chemical diagram, and that's what we have in our model. But then I talked about this, and there has to be management on that, which means that actually these simple arrows really have pools, separate pools, and transport mechanisms and other things all buried inside them. And so we wanted to begin to learn about the pools. So a question, if you have a model like this, is there 4P actually maybe on the plasma membrane? It's not on this diagram, but maybe there's a pool of 4P here that makes the PIP2, or do you have to wait for it to come from the Golgi? Is there a pool in the Golgi that actually contributes to the cell surface? So Eamon Dixon again asked that question. Where are the pools? And the answer he got, I'll show you before the evidence, was that there is a pool of PI4P on the plasma membrane that contributes to this pool of PIP2, and there is a pool of PI4P on the Golgi that also contributes to that pool. And before I go into his evidence, I want to also call your attention to an enzyme, which is here that he used. That's SAC1. SAC1 is a 4-phosphatase, which is actually part of the endoplasmic reticulum membrane. You could say, oh yeah, that's good to have a 4-phosphatase because you're not supposed to have any PI4P. So if any comes back there by vesicles, it just will get destroyed quickly. One of those guardian enzymes. But we use first an engineered form where just a little nugget of the catalytic domain is present. So it's not a membrane-bound enzyme. And we're going to do 2 experiments with that, and then we'll come back to the enzyme which is actually on the reticulum. So, and I have to introduce a tool called rapamycin dimerization, a tool that's very common for a cell biologist, namely to use 2 proteins, FKB and FRB, which have half a binding site for a drug rapamycin, and when you add the rapamycin, they come together around the rapamycin. So this is a dimerization system, and people have used it a lot for different transporting things together. In this case, a Lin motif is used to bristolate, palmitolate at the plasma membrane. So this piece is at the plasma membrane. So therefore, when they dimerize, that piece has to go to the plasma membrane. And on this FKB piece, you can put cargos. And in this case, an enzyme is put on here, and the enzyme is going to be that 4-phosphatase, SAC1, the little fragment. And so we are now taking the 4-phosphatase to the plasma membrane, and if it does something, then you have a pool of PI4P at the plasma membrane. That's the concept. Okay, so let's do that. When you transport it to the plasma membrane, here is time, and rapamycin is put on during this period to dimerize. And this red trace is a PrEP trace that says, yep, in 10 seconds, 15 seconds, the enzyme went to the plasma membrane. We have a pair of probes for that. And the ion channel says, okay, everything's good. And then, wow, something's happening to my PIP2 pool on the plasma membrane when I decrease that pool on the plasma membrane. So yes, there's a pool of PI4P on the plasma membrane, and it contributes to the 4,5P2 elsewhere. So then let's ask, is there a pool on Golgi and that contributes to the plasma membrane? Okay, so let's ask that question. So here is a picture where one cell, one TSA cell, has been outlined by me, and a circle has been put around where the Golgi is, and there is some gray stuff here we can hardly see, which is the phosphatase, which has color on it. And then an anchor has been expressed, which is a trans-Golgi network anchor, so that when you add rapamycin this enzyme should go to the trans-Golgi network. So let's see if that happens. So it's a movie. We'll add rapamycin in the middle. So my movie, yes it is. Yes, add rapamycin, and you saw the stuff go to the. So the enzyme goes to the Golgi. That's good. Does that hydrolyze PI4P? Well, we can express a pH domain, an indicator domain that likes PI4P, OSH, 1pH, and so it labels that actually likes Golgi PI4P. So it labels the Golgi 4P right there, and now it's the same cell, same time, different color. I will ask, does that pool get hydrolyzed? And the answer is seen. You add rapamycin, and the pool goes away, and so the probe leaves the Golgi. So the interesting question is, okay, what happened on the cell surface? Is this pool that's in the Golgi, does it actually contribute to the cell surface? And the answer is yes. Here is the threat trace that says the enzyme went to the Golgi, and this is the channel that says something happened to my PIP2 on the cell surface. That pool is disturbed by a Golgi depletion. Notice this takes only 20 or 30 seconds. So in 20 or 30 seconds, the channel PIP2 knows that the Golgi has changed. Now there's a remarkable rapid transport, traffic, could be vesicles, could be some exchange proteins. I mean, we don't know what it is, but we were all surprised about how fast it was. Okay, now we return to the SAC1 in its endogenous location, which is the endoplasmic reticulum. We're no longer using rapamycin. Oh yes, we are, for a different purpose. So here is the endoplasmic reticulum, diagrammatically. Here is SAC1, 2 transmembrane segments, and it hangs out in space. Before we were just using the little piece from here, but now we're using the whole thing. And people say that it hangs out in space enough to hydrolyze across on other membranes. So the question was, if we have a junction between the ER and the plasma membrane, will it hydrolyze that pool, and will it make a difference? Because this is now an endogenous molecule. So the way Eamon Dixon did this was he put on KBP on the endoplasmic reticulum and FRB on the plasma membrane, and you add the rapamycin, and they come together. And it works fine. You look in the turf microscope, you see them coming right up, brighten up. There are already many junctions between, as they're called, between ER and plasma membrane. There are quite a number of places where the turf footprint shows that. But now they considerably increase if you add the rapamycin in this kind of case. Okay, so the interesting question is, what about the channel? Is the channel influenced by that? And the answer is yes. So the channel says, you know, something happens by PIP2 pool. So the PIP2 pool here is affected by the fact that the endoplasmic reticulum is nearby. Now, so that implies that just organelle movements are going to act on changing the cell surface. Then Eamon went to molecules which are endogenous molecules rather than rapamycin in this system that are ones that make more junctions. You can overexpress these molecules, or you can put in antisense for the molecules and other things of that kind. And one of the molecules that is important in this is Pietro de Camilli has described something he called extended synaptotachyons. So it sounds like synaptotachyons is a synaptotachin, but it's related in some way, but it's a different class of molecules which make junctions between cells or between organelles. And it actually likes PIP2, so it makes a junction there because it likes PIP2. So when there's a lot of PIP2, the organelle comes up there and you start hydrolyzing PI4P and you have less PIP2 and that goes away. So it's kind of a thermostat which controls. Eamon showed also that you can make junctions with the ORI stim system. So if you make ICRAC for the depletion of intracellular stores, then we know that the endoplasmic reticulum goes to the cell surface, and Eamon found that then the PIP2 on the cell surface goes down as well. we showed that so I've been showing you that lipid homeostasis regulates, in this case, neuronal excitability dynamically. And so does the SNP1 and ethnicity and other effects. I'm sure there are many others. So I'm just telling you that we've kind of gone into the cell biology of what is it that makes this regulatory lipid and what might be dynamically controlling it in time. So, and that will turn off the channel, which will make you more alert for a while, and then put you back to sleep afterwards. So it remains for me to thank a whole bunch of people who worked on the Gproteincoupled receptors and have the jobs gone away, and the people who actually worked on the TIP2 questions, who are listed here. Byung-Chong Seo was the one who originally discovered it in 2002. Jill Jensen did her thesis on fast kinetics, and is still with us. Jordan Falkenberger and Eamon Dixon did more kinetics of the IP3, calcium, lipid synthesis, lipid breakdown components. And Eamon also did the work I just talked about the movements of SAC1. Bjorn and Martin Kruse and Oscar Vivas have recently taken the work that we had done so much in TSA cells, the expression system, and brought it back to the sympathetic ganglion. And they are now beginning to publish their work on doing the same kind of experiments with very similar results and describing the kinetic basis of the kinetic properties of the neuron of these fields. And Wupan Day was the one who expressed the SNP1 and showed an increase of groups. Thank you very much. It wasn't quite an hour.
Any questions, comments?
Yeah, let's start with students. Larry's old. Larry's a student, yeah. Okay. You're a postdoc.
So it seems like the PI, P1, PIP2 is really well described in how it affects physiology. So I was wondering do you know if there's enough PIP3 phosphatase activity to affect the available PIP2 pool in any way that produces regulation of ion channels?
So the PIP3 phosphatase you mentioned, that would actually make PIP2.
Right.
But according to Lou Cantley, when he was originally discovering the PIP3 and PI3 kinase system, that those phosphoinositides are 1% lower than the PIP2. That's what he said. Which means that if you're making PIP3, according to that, you just take 1% of it and then you're done. But I think it must be more, and that because you can actually locate it with pH domain indicators, and you couldn't see 1% of the PIP2 that way. So I think we don't actually know. We should have tuned our mass spec to simultaneously measure PIP3 and had a better idea. We don't actually have good measurements of that. Do you have your question, more or less?
Yeah.
And any students? An MD PhD student.
So,
7th year.
9th year.
9th year.
So,
It's a whole decade.
we'll square it off.
Graduate school.
The endolysosome experiment, have you tried that with a KV channel that does not require PIP2 and do you see activity?
That's a good idea. No, we have not tried it with anything else. It's kind of just an isolated small experiment. We'll continue, but I'm just showing you a preview of where we might go. It's a good idea, though. So we would pick one of the several KV channels which do not respond to the BSP and so forth. Okay? Yes.
My first question is, we always have this question, how much PIP2 are you? Do you have any?
So the question people may not have heard was, how much PIP2 is there in the plasma?
Yeah, like how many percentage do you think PIP2 really like normally?
Yeah, so it's certainly less than 1% of all the lipids. It's more than 1/10 of a percent. So it's somewhere down there. So it's a low, it's 0.5% of the total lipid or something like that, I would say.
And then the second question is, so there are many membrane proteins that are binding to PIP2 and they get regulated. So have you thought about that the binding affinity of different proteins
Yes.
may also...
Okay, very good question. There are many proteins binding to PIP2, as you say, and have we thought about the binding affinity? Yes. The literature on channels, for example, already shows that some channels have high affinity for the PIP2 that they like, and some of them have low affinity for the PIP2 that they like. And the high affinity ones can remain with PIP2 even if you deplete PIP2 a lot, and you have to work very hard to show that they like it. And the other ones respond right away. And there are many proteins, yes, so when we made a model, we had to sort of settle what are the pools of PIP2 that we were going to put in the model. And it turned out in the end that we had to have a pool of free PIP2, which was half as big as a pool of bound PIP2s. So there's 1, 2, 3, 2 bounds for every 1 free. And the bound ones would be the ones on proteins that like PIP2. There's many more PIP2s free even than there are KCQ channels, even when we overexpress them. So we haven't used up the PIP2 by expressing the channel. There are lots of partners out there that like it. Stuart McLaughlin also measured the fluorescence. What is this? The technique where you measure motion particles with fluorescence and correlation spectroscopy, I guess it is. The mobility of PIP2 and came up with the concept that it was about 1/3 as mobile as it ought to be, and which is supporting the idea that 2/3 of it is bound and 1/3 is free. More.
So, according to the Hille hypothesis, PIP2 is required to keep the channel shut off during trafficking. Right?
The absence of PIP2.
Yeah, absence of PIP2. So then, according to that hypothesis, every channel should be interacting with PIP2.
So that would be the, I mean, you
turn
might
it
say,
on.
of course, I mean, let's say we believe the hypothesis. That would be okay if every channel was bad for the other organelles. But maybe many of the channels are okay. Maybe, some potassium channels are fine, and they don't bother what the organelles do. And so there's no selection pressure for them to keep a PIP2 sensitivity for that reason. That's what I'm thinking. Or maybe the channel is good for 3 compartments and not for others. So then it would be forgiving. It would like PIP2 and something else instead of just PIP2. So when we do the experiment with VSP, which takes PIP2 and puts it into PI4P, you might say, well, PI4P, maybe it likes PI4P too, also, if it doesn't decrease. And if it does decrease, then you can say it doesn't like PI4P because I just made PI4P and it turned off. So the KCQ channel turns off, doesn't like PI4P, but other channels don't turn off. Maybe they are okay with PI4P. So we don't know the scope yet of selectivity, and I think some membrane proteins are okay in different cell membranes, and there's no reason to bother turning them off.
Some similar.
Now Larry takes precedence.
Okay, so,
Are you okay?
yeah,
Good.
I'll recover.
Maybe you can ask during the dinner time.
Well, we're gonna have a lot of time.
Ask other students. I can always, I'll ask you later.
Well, you can. If we have more students, yes.
So I have a question about.
It's good, when one student asks a question, then the other students get brave.
Yeah.
So I have a question about the model with the dose response. I'm not exactly sure what you guys. I assume some kind of just a cascade, so you can get amplification and shifting of the doses.
Yes.
But then later you're talking about how the Golgi and the membrane population can be kind of shifting the population of PIP2 and be sensitive
Right,
to that.
yes.
So then the model isn't quite.
The model isn't quite right.
It isn't quite
because
right.
It doesn't have a transport component.
Right,
and
you
it
need
doesn't have
different
multiple
kinetics.
compartments in it.
So could that, I was wondering, can you modify the model to actually explain how come the Golgi can have such a fast kinetic on kind of how
I think
fast
first
it
we
is?
have to do biology. I mean, the first thing is to find out what process is it? Is it vesicles?
Mhm.
Is it an exchange protein that's transferring them? Because maybe it's vesicular traffic. None of us is used to the idea that vesicular traffic is that fast.
Mhm.
But I think we have to do the biology before we make the model. So the model,
Right.
model should follow after some knowledge about what the components are going to be.
Mhm. So do you have any
That's my
idea what
approach.
it might be? Like, any,
I don't. No, I could hint to you, but I can't because I don't have any human hands. More students? Are you a student?
Oh, no.
And gentlemen.
Okay. So is there any like a local concentration of PIP2 that is in the membrane that is different? It's not homogeneous. It's
Yeah. So
yeah.
are there,
And does that affect the measurement that it
Yeah. So that's a vexing problem. You know, for example, among other things, people might say, well, you activate a receptor in phospholipase C, so there should be a gradient of PIP2 right there. Of course, you depleted the PIP2 there, but not elsewhere. And Don Hilgeman says, well, you know, it only takes 1 second for the stuff to move over the whole cell surface, so I can't sustain the gradient, he says. He writes papers about that. Now, you can have a gradient if you have a line which is a corral or a fence, so that you can't get diffusion across things. And in some cases, like the so-called immune synapse, where an immune cell comes and attaches to another cell, then in this compartment, which is part of the original immune cell, there is a completely different composition of phosphoinositide, which doesn't seem to diffuse around that little edge, as if like a patch, you know, a patch clamp. So, okay, and your question was, are there patches? So there are probably patches. So people talk about rafts. Rafts is sometimes not, I mean, some people are polarized about rafts. So you don't have to use the word rafts, but there are obviously regions where different proteins and lipids are together. And PIP2 might be enriched in some of those, so that and with some channels there, so that there's some special compartment. We use the pH domains as an indicator of phosphoinositides. So you have pH domains are just domains that like and recognize head groups of phosphoinositides. So they would go to the cell surface. And when we add ligand to a whole cell and there's pH domain inside, it leaves every part of the cell without apparently saying that there's part of the cell that's immune to the receptor action and part of the cell that isn't. So there's nothing very patchy about what we see. Then you could say, okay, well, your microscope is a light microscope. In fact, it's a patch clamp microscope, so you can't see very well. And with the high resolution, maybe you'd see better. And that might be true. That maybe the raft is 100 nanometers, and we can't see it, or 50 nanometers, and it just contains a few thousand lipids. Those are all possibilities which we can't dismiss. And then, of course, modeling that would require some understanding of whether you can just lump that all as one thing or whether you have to consider those separately. Is the receptor there? Well, the receptor seems to take away all the PIP2. So the receptor reaches all the lipids. And interestingly enough, you notice that the PI4P went down a lot. That was with a plasma membrane activation of a receptor. But the earlier diagram said that a lot of PI4P is inside the cell. So that means almost all the PI4P visits the cell surface every 60 seconds, which is how long that was, and gets hyperlyzed. That sort of gives a limit to how compartmentalized they can be.
So you have this whole collection of channels regulated by PIP2.
Yeah.
Big collection of channels, and you would think that there should be a highly conserved structural domain that interacts with PIP2. That's
Yes.
highly conserved and present in all of And if that's not true, then it means that by convergent evolution, they all develop different places that interact with PIP2.
Yeah.
But that seems, why PIP2? Why are they all interacting with PIP2 if the
Right.
site isn't conserved?
Yeah, okay, so you were arguing against convergent evolution. I'll just argue why there is pressure for convergent evolution, adaptive pressure, which is that maybe each of the channels that likes PIP2 is bad for other compartments. And PIP2 is a definer of the cell surface, and there aren't so many other molecules that define each of the membranes as nicely as PIP2 does. So all of them will find some evolutionary pressure to admire PIP2 and respect it. And so improvements of that system will be selected for. So that would be a sort of convergent evolution. My colleagues here seem to say that all the known PIP2 binding sites on KV channels are the same, that they all map to the same things. But tell me, how does it map the KV channel onto the KIR channels?
Exactly the same.
Same as well?
Same.
Okay, so
Same.
the KIR
With
channel.
respect to the pore. Same location, it's not the same structure.
Correct.
Not the same sequence.
But assuming the conserved structural feature is the pore, then you could have a divergent evolution leading to the loss in certain channels rather than convergent.
Colin, did you want to add to this discussion?
I think you just need positive charges in the area, so it's not,
Yes.
it's sort of an induced fit. because
People
it's
talk
such
about polybasic regions.
Yes, because it's such an electronegative molecule.
Some
That's
people
what's
would like
striking.
a similarity to pH domains, which are other domains that like. There's nothing that suggests they're like pH domains in terms of pH domain is defined as a fold. But in the pH domain, it's 100 and something amino acids, and the collection of positive charges comes from different parts, and they all come together. So it's not like there's a little nugget that's just right. So there is the pH domains are on enzymes that, like the AP2 adapter for collagen, and like the dynamin, and like the synaptogenin. Those are things that recognize PIP2 and use it as a location for their actions.
But when we talk about the PIP2 regulation of the channels, we're talking about some channels that are absolutely dependent on PIP2, and there are only a few places in the channel that can be affected by PIP2 that completely turn on, turn off the channel. So I think that also has some pressure too.
I see. Yeah, even if you try another scheme, they're all going to be so close together they'll look like the same thing in the end.
Yeah.
Right? Maybe we're getting off the subject. Does somebody want to?
Speak to follow up on that question about PIP2 domain or patches in the membrane.
Yes.
For when you did the experiment where you see the ER moving towards the plasma membrane,
Yeah.
that was in TSA cells, which are flat.
We've also shown the same in neurons.
Okay, do you think it's a universal thing for all cell types? And
SAC1 is there in yeast. That's where it got its name from. And so, and it hangs out in yeast as well. So I think it would extend to yeast, for example.
do you think something like a cardiomyocyte where you have like huge...
Cardiomyocytes more closely related to TSA cells than to yeast. When I say cardiomyocytes, must have the same. Now, the ER, you're talking about SR. We would talk about SR in the cardiac ventricle. Is the SR like the ER? Well, of course, it's somewhat like it, but how much like it? And I didn't even know whether enzymes like SAC1 are on that or whether they're on a separate component of ER. Cardiomyocyte is sort of an embryonic name, I guess. Is that right? Or is it the name of an adult? I guess
Like an adult.
Okay. So where would the endoplasmic reticulum be other than in the SR? There must be a place where they make proteins that has ribosome.
I mean, like, morphologically, how far do you think the ER can move in a cell to get close to the plasma membrane?
Yeah, I suppose it has to be pretty close. I mean, the extended synaptogenin synaptogenic, has a binding site for PIP2. And it doesn't go to the plasma membrane. It adheres to the plasma membrane when it bumps into it. So it has to be able to just go spontaneously back and forth and sample whether it's there. It's not going to be direct, at least we don't think it's directed or propelled to the cell surface. So therefore, the structured transverse tubule SR structure of a real muscle cell probably doesn't admit any of that kind of stuff. It has to be something else that's surface cisterna or some other more loosely bound part. Now, is Cohen, did he leave? Oh, he was going to ask a question. Then maybe we can let people go.
Any other questions?
There,
or
whatever.
comments? We need to get back to cardiovascular and cardiac.
we did sort of have a cardiac question.
A little bit.
You want to ask a cardiac?
And is there anything known about PIP2 role in gap junctions and connection 43 and so forth? If there is an organization, spatial organization of PIP2's distribution in the membrane.
I think it's known about gap junctions. As far as I know, there's no.
Because there is a lot known about trafficking of connection 43 to the intermediaries. And I was wondering if there's any association.
So the famous Don Hille, who discovered the sensitivity in cardiac cells, then made a HPLC system to analyze the lipid and to ask, does the lipid get decreased in the cardiac cell? And it doesn't. We couldn't find any evidence that decreased the lipid. The lipid is made as fast as you want to make it. It's a transcript. It's synthesized as fast as you make IP3. So then he was disappointed and he sort of abandoned his hypothesis. And he was surprised when we came into the neuron and said it gets depleted. And then he said, I don't believe it. So we went to Don first with our TSA cells and measured it there, and it did deplete. And then today I showed you our own measurements on the neurons.
I saw the measurements. But the interesting thing is that the intercalated disc is such a hub of expression of many. Sodium channels are there.
Interesting.
Connection is there. And I wonder if there is any, but if nothing is known, then maybe it's a good place to look.
It's a good thesis project. Or beyond. I said it's a good thesis project.